All right, I guess that's the cue to get going. Thank you everyone for being here this morning and to help me kick off this session on biogeography. My name is Jonathan Rader. I'm a postdoc in Daniel Matute's lab at UNC Chapel Hill. And I'm going to talk a little bit about a project that I've been working on for a little while that I think is finally starting to bear some pretty interesting fruit. And I want to begin by noting that one of the most striking patterns in global biodiversity is the observation that pretty much every group of organisms is most diverse near the equator in the tropics, and that biodiversity diminishes as you move outward toward the poles. To illustrate what I'm talking about, I'm showing you species richness here from Buckley et al. 2010. Notice the pronounced band of warm colors across the tropics denoting high species richness. Also note how that contrasts with the cooler colors and lower species richness in the temperate regions. Now, this isn't really a recent observation. The latitudinal biodiversity gradient was noticed long ago. So today we're standing on the shoulders of some little-known figures, people like Humboldt, Wallace, Darwin. But the latitudinal diversity gradient has been the subject of increasing recent attention. Here I'm just showing you a small smattering of some of the recent papers on the topic, but there are hundreds more. And several explanations have been proposed to describe the latitudinal diversity gradient, and most of these are focused on the geological or the climatic setting. And that makes some pretty good sense because there are obvious climatic gradients in temperature, humidity, rainfall, et cetera, and these could be drivers easily of a biodiversity gradient. There was also historically more land area distributed in the tropics, providing greater opportunity for tropical lineages to emerge. And this is augmented by the fact that the tropics were just latitudinally broader in the past. These factors, though, are exogenous to the organisms themselves, like this example that I'm showing you here that describe the influence of climatic fluctuations on the diversity gradient. These explanations, though, imply that the latitudinal diversity gradient is a byproduct of external factors. But that might not be a complete picture. Endogenous factors, such as endothermy, also influence the strength and distribution of the diversity gradient across latitudes. And also, this gradient might be the result of evolutionary processes. There are a couple of hypotheses that offer evolutionary explanations too. One of these is the tropical niche conservatism hypothesis, which proposes that the latitudinal diversity gradient is the result of most taxa originating in the tropics. This could be because of either high diversity rates in the tropics, longer evolutionary histories there, or the greater land area in the tropics. And I'd like to note, none of these explanations are exclusive of each other. But when paired with relatively low dispersal rates to more temperate regions, this leads to an accumulation of taxa in the tropics. And there is some support for this idea. For example, Kirchhoff showed that most angiosperm taxa emerged in the tropics, and that evolutionary transitions, which you can see here in the arrows in this figure, evolutionary transitions toward more temperate regions have been comparatively rare. Another possible explanation is the out of the tropics hypothesis, which suggests that taxa preferentially originate in the tropics, but then expand outward toward the poles, and that diversity is simply a function of distance from the point of origin. Both of these hypotheses have some support, but we have lingering questions. First, to my mind, is which of these hypotheses, the tropical niche conservatism hypothesis or the out of the tropics hypothesis, best describes the evolutionary patterns underlying the latitudinal diversity gradient? And second, do we get different results if we look at different groups of organisms? To answer these questions, we're leveraging the newfound wealth of phylogenetic information. The abundance of well-resolved, large-scale phylogenies is a massive boon to these kinds of studies. We're also drawing from the tremendous amount of biogeographic data available in databases like the Global Biodiversity Information Facility, or GBIF. GBIF has almost 3 billion individual observations of animals, plants, fungi, et cetera. So we're working with the occurrence data for several vertebrate groups: birds, mammals, squamates, amphibians, also angiosperms, fungi, and several major groups of insects. So here I'm showing you a big, ugly plot of all of the observations in GBIF for mammals. And I've got two first impressions. First, holy crap, that's a bunch of data. And yeesh, not all of it's good. We have some weird things happening up here in the North Pacific. We've got a whole line of things that are just stuck right along the equator. So we've done some filtering. And we filtered to remove duplicates, suspicious observations, things that were tagged at like zoos or museums or things like that. We've also removed things that are stuck at arbitrary locations, like the intersection between the equator and the prime meridian. For this project, we've also restricted our focus to terrestrial habitats. We've filtered the data to include only taxa that are represented in their respective phylogenies. And we've also excluded species for which there are fewer than 20 observations in GBIF. And at the end of all of that, we get something that looks a little bit more reasonable. Again, I'm only showing you mammals, but we've done this for all of the groups that I'm going to talk about today. Once we finished filtering the observation data, we used the remaining observations to estimate the latitudinal range for each of the species. We took the middle 95% quantile range as the latitudinal range and the median as its centroid. We used these latitudinal ranges to calculate an index of tropicality for each species by adapting a method pioneered by Kirchhoff, and one which our lab has used previously in a study on sandflies. This method considers the proportion of an organism's latitudinal range that exists within the tropics. The tropicality index ranges from -1 to +1, and it's centered around 0. 0 indicates that the species' latitudinal range is split evenly between the tropics and temperate latitudes. When the tropicality index is -1, or is +1 rather, it indicates that the entirety of the species range is contained within the tropics. This is a tropical species. And when the tropicality index is -1, it indicates that no part of the species range exists within the tropics. It's a fully temperate species. So we calculated this tropicality index for more than 40,000 species. In order to describe evolutionary transitions in tropicality, we binned species by their tropicality index into 4 states: temperate, semi-temperate, tropical, and semi-tropical. Again, this followed the methodology of Kirchhoff et al. Here I'm showing you the phylogenies for mammals, squamates, and angiosperms. We've mapped the tropicality index onto the tips of our phylogenies. Tropical species are in red, temperate species are in blue. And I know there's a lot here, but the main point that I want you to take away from this is that there's a lot of variation in tropicality among these lineages, and this is representative of all of the groups that we've looked at so far. The ancestral character state reconstructions allowed us to quantify the evolutionary, so we conducted ancestral character state reconstruction that allowed us to quantify the evolutionary transitions among tropicality states, which we can represent here on these transition matrices. I want to orient you here a little bit, since we'll be using these matrices to illustrate our results. We have the 4 ancestral states here across the x-axis, and we have the 4 descendant states on the y. Right? Each of the cells depicts the number of transitions between nodes and tips of the tree. I've color-coded these to make any patterns that emerge a little bit easier to see. Darker red denotes more transitions in that cell. As a reminder, we're testing 2 hypotheses: the tropical niche conservatism hypothesis, which predicts that taxa originate in the tropics and that dispersal is low, and the out of the tropics hypothesis, which predicts that taxa originate in the tropics and disperse outward toward the poles. With that in mind, we can make some predictions about the patterns that we might expect to see in our transition matrices under each of these regimes. A strong diagonal, such as this, showing that taxa tend to remain in their ancestral state, would be evidence for tropical niche conservatism. On the other hand, if we see the bulk of transitions shifted to the right on the matrix, it would suggest that taxa are transitioning away from the tropics, supporting the out of the tropics model. Okay, so now that we have some predictions, let's look at the data. I'm going to start by showing you the transition matrix for mammals. This represents almost 10,000 species of mammals and more than 20 million global observations. And there are 2 patterns that emerge. First, we see that strong diagonal indicating that descendants resemble the tropicality of their ancestors. So mammals are showing some evidence supporting the tropical niche conservatism hypothesis. But the keen observers out there might have noticed something else too. There's also a large number of transitions from all of the subtropical states back toward the tropics. This doesn't fit with either of our predictions, and I'm still not entirely sure what to make of it. But we have other taxa to look at, so let's work through those. Here are the data for squamates, lizards, snakes, and things like that. And again, we see the strong diagonal. But we also see the pattern of subtropical to tropical transitions. And we see the same thing for amphibians, too. So this is starting to look like a pretty pervasive thing. But so far, we've only looked at vertebrates. So let's take a look at plants. Here are the angiosperms. The diagonal pattern here is especially strong, which I find to be refreshing because essentially we've replicated the results from Kirchhoff with different data and a slightly different analytical method. So this provides a nice sanity check. But if you squint at it, you can kind of see the subtropical to tropical pattern here too. So overall, I think we're starting to see some general support for the ubiquity of the tropical niche conservatism hypothesis, along with some other interesting avenues for further study. So to wrap this up, and hopefully in time for some questions, we've used an absolutely massive observation database for more than 40,000 species to test between two hypotheses explaining the latitudinal diversity gradient. We found strong support for the tropical niche conservatism hypothesis. Most lineages tend to remain in the climatic regime in which they originated. By contrast, we found little support for the out of the tropics hypothesis. But we did also uncover an unexpected pattern of lineages transitioning from subtropical regions back to the tropics. It appears to be a pretty pervasive thing, and I still haven't fully wrapped my head around it, so I would welcome any thoughts that you all have on that subject. Looking forward, we're working on some simulations to hopefully help us better understand those subtropical to tropical transitions, and if nothing else, to give us some confidence that that pattern is not simply an artifact of methodology. We're also adding more taxa to the analysis. Any major group for which there's a well-resolved phylogeny and sufficient observational data is welcome. We're already looking at fungi and several groups of insects, including butterflies and bees, and we're also happy to accept suggestions from the audience. With that, I have many people to thank, and especially all of you, and to all of you for being here, and I think I have time for a few questions. Thanks.
So you're only dealing with extant taxa. If there's a tendency for tropical lineages to have a higher extinction rate, might that explain this apparent tropical movement?
Yeah, I think it might. And so only being able to work with the extant taxa is obviously a limitation of the study and studies of this kind. And this is exactly why we're conducting these simulations, is to try to understand under what sort of evolutionary regimes we would expect these kinds of patterns to emerge. Yeah, I think.
Yeah, thank you for the talk.
Thanks.
I'm Theo from the University of Lausanne. I think when you talk about that transition from subtropical to tropical that you observe, you talk about latitudinal range, but as you mentioned at the start, the tropics were much wider at the start. So tropical species more like something of a climatic niche, right?
Right.
So a transition from subtropical to tropical is actually maybe just staying tropical, but just moving, like shifting range to like track the climatic niche, which is also like really consistent with tropical niche conservatism, right?
Yeah. Yep. Again, we're hoping to be able to simulate exactly that, the shifting of the tropical band, and to work that into the more sophisticated models that we'll present in the manuscript.
Actually, two questions. So first,
Sure.
Did you use any fossils to help inform anything for the ancestral state reconstructions? And if not, do you plan to try to incorporate those into certain taxa?
We didn't do that personally, but I think that, I'm trying to remember, I think most of the phylogenies that we're working with are time calibrated with fossils. So that's kind of implicitly built in.
And then my other question is that, so the out of the tropics model is of course built on marine taxonomy biomes.
Right.
Predicated on that. Do you think, and there's some indications that at least hotspots might be formed differently in marine versus terrestrial species. So do you think that if you included any sort of marine taxa that you might see different results, or do you have any plans in the future to incorporate the marine taxa?
This is probably my own personal bias here because I come from a background working with birds, and I'm moonlighting with insects right now. So I think that's why I sort of gravitated toward tropical or toward terrestrial habitats at this point. But yeah, I do believe that we would see different patterns if we look at. I got the gong. I do think that we would see some different patterns if we look at marine taxa as well, and I would love to be able to do that in the future. I'm not sure if that's going to be part of this project though. But yeah, it's a great suggestion. Thanks. Great.
So thanks for coming. I'm really excited to be here today. My name is Chad Eliason. I'm a research scientist at the Field Museum. I'm going to be trying to answer this, providing an answer for this question to you. This has kept me up at night, maybe not everyone in this room, but hopefully you'll see some cool pictures of some really colorful birds and get some ideas, or maybe give me some ideas on what these results might mean. I'm really glad I followed Jonathan too, because he gave a really good introduction on latitudinal diversity gradients. So I'm not going to really go into that much. I was kind of playing around with some PowerPoint AI here with these cool little bubbles. But anyway, the idea of this slide here is a lot of us in this room, in this conference, have thought about these diversity gradients, whether it's in the number of species or other axes of biodiversity like phenotypic diversity, such as body size. There's this cool study I found maybe coming out soon on availability of reference genomes and this kind of latitudinal diversity gradient, or just a latitudinal gradient there. But also in the upper right there, this idea of colorfulness showing this same trend. And this has been more recently kind of studied by some researchers. So there was a study that came out, I'm not sure if Chris is in the room, but Chris Cooney and et al. A couple of years ago showed this really cool paper, pretty much across all birds, I believe, this gradient of more colorful species in the tropics. This was interesting, but I also thought, there's this paper in 2015 that kind of showed that birds, butterflies, and some other groups, they do not show this gradient in coloration. So there's kind of this it's not a debate, I guess. There's just some differences, I think. Maybe it could be the clades that we're looking at, the areas we're looking at could be clade specific. It could also be, I think, mechanism. I think mechanism might actually be important when we're thinking about these gradients in color, because there are different ways animals can produce color, and we might have reasons to think that different mechanisms evolve in different ways. So for example, the study in the 70s by Bailey showed that in North American birds only, there was a gradient in different forms of coloration, specifically iridescent coloration. And I'll show you what this looks like in a second. So that's kind of the idea of this talk here and of this study, is that color mechanism might be important in understanding diversity gradients. So here we have an electromagnetic spectrum. I've showed this many times. Hopefully you haven't already seen this if you've ever seen a talk I've given. But birds produce colors either with pigments in their feathers to give some of these yellow, orange, and red colors. But they can also produce colors by not eating or producing a pigment, but producing a structure in their feather that causes what's called a structural color. I always tell people, and hopefully not many people do it, but if you get a feather, like a peacock feather from like a craft store and destroyed it with a hammer, the color would go away and you'd be left with black or brown melanin pigment because you destroyed the structure to produce these really brilliant iridescent colors. What I want you to see with this slide, though, is that the color gamut, or kind of the spread of diversity of structural colors, is much wider than pigment-based colors. So this was hypothesized by Cassie Stoddard in, I think, 2011 now. This idea that different color mechanisms, specifically structural colors, could allow birds to produce a much wider range of colors than other mechanisms like pigment-based colors. I'm not going to go into real depth on how these are produced, but here I just wanted to show this Anna's hummingbird, just to give you an idea of what iridescence looks like if you haven't kind of seen this change in color with angle. This is actually described by Isaac Newton. I think there's a 1700 paper I like to cite, first talking about iridescence. It's cool to cite old things, I think, sometimes, and also useful. But in this case, you can kind of see the green-red shift. You can also see kind of color going from pink to black. In the upper left here is this, the Cornell Lab of Ornithology put this diagram together of just kind of showing how this is produced. So as you change the viewing angle, you're changing the distance that light has to travel through these structures. The black there is just melanin pigment, same stuff in our hair, our skin. The gray is just layers of keratin protein. So structural colors we know that they evolve differently at different rates, and they kind of have these unique evolutionary dynamics compared to pigment-based colors. So the idea is whether this has an effect on the latitudinal gradient that we might see in color, and whether it exists for structural colors in the first place. Another cool thing about structural color, I think, is that there's a direct link to the environment with structural color as compared to pigment-based color. So the structures that cause these bright colors often result in trade-offs, like water repellency. So if you have a bright iridescent feather like this duck on the left, can people hear me all right? I just realized I'm a little too tall. Okay. They have lower water repellency. In the center here, I'm going to show a video here of me changing humidity rapidly from 20 to 80%, kind of instantaneously. You can see this mourning dove feather kind of changes in color. Hopefully you saw that going from like a pink to a bluish purple or violet, and then back to pink as it dries out. So these are kind of instantaneous changes. There's also relationships with kind of the thermal properties of feathers. So iridescent feathers warm up and heat up faster. So there's these really cool links with the environment that might be kind of tell us that we should be looking at these gradients and trying to understand them. And I think also it's really important to kind of consider the whole thing. So geography, speciation dynamics, and mechanisms. And this is kind of the crux of this study here, of this idea of what I'm going to be showing now. So to kind of understand, to tease apart these three things and how they interact to produce latitudinal gradients and coloration, we first needed a database of structural color and different color mechanisms in birds. So here are the three different kinds of forms of color that we collected, data we collected. So non-structural or pigment-based colors can be produced anywhere in a feather. I just have a little cartoon of a feather there. Iridescent colors, like in that hummingbird, are found in the smallest parts, the feather barbules. It doesn't really matter for this talk. But if you have questions about that, I'm happy to talk later. And then non-iridescent structural colors are another form of structural color, but those are produced in the barbs, kind of those yellow parts of the feather. So how did we do this? We first started with photos and drawings. So we went to the Handbook of the Birds of the World, looked for photos. We went out, looked at our 10,000 bird species, tried to score presence or absence of these different forms of color. If we ran into cases such as like a folded wing painting where we couldn't tell if there was iridescence under the wing, we went to museums. So we looked at 6,000 to 8,000, I think, different species in museums collections. In some cases, we were still a little bit puzzled by what the color mechanism was, so we used microscope images to kind of tease that apart. So here are just some zoom-ins of these feather structures. The cool thing is we did around 10,000 species, but we made about 35,000 scores by individual readers. So we had been building up these databases on our own, and we decided let's combine them all. So we can look at consistency across reading and things like that. So we scored each species about 3 to 4 times by different people. So what did we find then with this gradient? Does it exist in structural color? So yes, it does. If you kind of squint, I kind of did these pies, but it doesn't show it maybe as strikingly, but it was a good way to show all the data. But we do find that there are proportionally more structurally colored species in the tropics. If you look at the pie charts in the lower left, that's kind of the summary of overall. So there are more iridescent species, 1,300 compared to 324, and also the non-iridescent structurally colored species, 916 to 180. Okay, so we use chi-squared tests and some, kind of more basic statistics to kind of calculate that this gradient exists, but why might it exist? What are the mechanisms, the evolutionary mechanisms that might have caused this buildup of structurally colored species in the tropics? So here's where we came up with 3 different ideas, 3 different hypotheses. First one could be that potentially species that have iridescent or structural color are more likely to disperse or expand their ranges over evolutionary time into the tropics. And this would be, kind of outweighed by maybe the shift in pigment-based colors. So if you have this kind of unidirectional change or in only structurally colored species, that would cause a buildup over time. Second, it could be that you maybe evolve structural color in situ in the tropics, more likely then lose it, going back to the pigment-based form. The third hypothesis, is that maybe structurally colored species speciate faster in the tropics. And there is some support for this in smaller groups of birds, and I'll talk about this a little bit in a second. So what we did with all this data, we had a tree, we had geographic data. This paper, actually, I got the timing pretty well here. It came out yesterday, so if you want to take a look at that, it's out now. The database is out if you're interested in looking at that. We needed a big model to understand kind of these transitions. So really the only thing I want you to see in this model, don't worry about all the parameters, just the idea is that it's one of these, it's a cladogenetic, state-specific speciation and extinction, or a classy model, fit in diversity and looking at rev bays and things like that as well. But basically the gray lines just show like how a species can move around, so from an ancestor. So let's say you start up at upper left there, a gray line shows you could move from kind of the yellow color to the blue color, which means you had non-iridescent structural color and you lost it. So with all these things, all these parameters estimated, we can then look at those relative support for those 3 hypotheses that we had. So now I'm going to show the results of those 3 hypotheses. First off, we find that iridescent species disperse or expand their ranges to the tropics faster. So here we have a parameter distribution of the net dispersal direction to the tropics. We find support for that more strongly for iridescent species, which we think is pretty interesting because there is a lot of kind of genetic studies out there showing pleiotropy in the melanocortin system. So a lot of melanin often leads to things like aggressive behavior or dispersal behavior. There are some of these links between behavior and melanin pigment at kind of the individual species level. It could also be that species, there's a lot of things included in this at a macroevolutionary level. It could just be that species that are iridescent, when they expanded their ranges in the tropics, having a rare or novel color in one of these dense rainforest communities could have allowed them to kind of persist in that environment. So we're trying to understand, maybe teasing apart the biology of this trend. With respect to the second idea, the tropical transitions, we find some support for this idea, but only in the non-iridescent structural colored forms. So iridescence was not gained faster in the tropics, but the non-iridescent structural colored forms were. There is some work out there showing that changes in molt speed can affect the ability to produce these kinds of colors. So it could be that maybe a slower pace of life in the tropics is part of the reason for this. Again, it's just a little bit hand-wavy. The biology of it will need to be borne out with future work. But as far as what explains this overall gradient, this was the second factor that did. We didn't really find much evidence for this idea. So this was shown in African starlings that species with novel colors, iridescent colors, diversified faster than ones with more drab or boring colors, I guess. We didn't find much support for this, and it could be that we're just looking at a very coarse scale and also a very broad scale. So again, I think looking at clade by clade, these different kind of patterns could be really interesting to kind of look at the fractal nature of evolution. We also did some ancestral state reconstruction. So RevBayes has this ability to kind of do this stochastic character mapping with these really complex kind of classy models. Again, if you look here, a lot of gains. There are 302 gains, 415 gains of iridescence. The really cool thing, I think, is that the ancestral state here of the whole tree, so the ancestral crown bird, we recover as very strongly supported as iridescent. I think this is exciting. That we have this modern kind of neontological approach that kind of, I think, reflects. There's a lot of paleontological work. I've done a little bit of, I've dabbled in this a little bit, but we have a lot of evidence of stem birds and other dinosaurs that are closely related having iridescent forms of coloration based on the structures that can be preserved in the fossil record. So I think this is really cool. Some conclusions and next steps. One thing, I showed different macroevolutionary dynamics for different color mechanisms. I think this just tells me that we should probably be taking mechanism into account in our evolutionary analyses, as I know a lot of us in this room do. The buildup of structurally colored species in the tropics being driven by dispersal and in situ origins, I think tells us that the next steps are we need to tease apart whether it's intrinsic things like pleiotropy or is it extrinsic factors like sexual selection and ecology that causes this. The early birds being iridescent, again, I think we can and maybe should start to look for more iridescence in the fossil record. And now that this color database is out, I think, or I hope that it will fuel further research on structural color evolution in birds. I just want to end with this slide here, just showing kind of one of the little springboard projects here. So I've been working on this grant-supported project with collaborators on this really colorful, cool group of cute birds, these Todiramphus kingfishers, found across the Indo-Pacific. This is some of their colors using spectral reflectance meters. This is kind of a springboard for some of these ideas, and it's also kind of this ongoing project that we have. And I just want to do a shameless plug here of a few studies, including the next talk coming up right now, Fernando's talk. You should stay and watch. And then if you want to see a little bit about the diversification and kind of the phylogeny, the phylogenetics of this group, Jenna is giving a talk on Sunday there. So with that, thank you, and if there's any questions, I don't know.
Hi, my name is Fernando. I'm a postdoc in the University of New Mexico. Thank you, Chad. I'm so glad I'm presenting. Thank you for being here. I'm so glad to be presenting after Chad and Jonathan, because I'm going to talk about conservatism, niche conservatism, and birds. Those little guys there. Okay, so let's do this. I am a biogeographer, and in general, I'm interested in understanding how large-scale conditions have shaped the history and the distribution of lineages broadly. My background is in birds, but of course I've been influenced by giants and one fundamental concept of my research has been the fundamental niche. So we can define the niche as the set or the range of conditions in which a species can survive and maintain populations. This is based on Hutchinson, and of course this is without the effect of biotic interactions. So here I'm just showing two abiotic large-scale conditions, and the niche is usually represented as an ellipse or as an ellipsoid if we think on the n-dimensional hypervolume of Hutchinson. And within the niche, the growth of populations is positive, R is positive, the intrinsic growth rate. And outside the niche is negative. So if we see this from a population dynamics perspective, we can think in the environmental space as source populations being inside the fundamental niche and sink populations being outside the niche. And from this fact, which was shown by Holt and Gaines in 92, but also kind of exploited by Pulliam in 2000, we can see that most individuals of a species will be found within the fundamental niche. This creates a bias, as Holt and Gaines pointed out, and this bias is probably one of the causes of niche conservatism. Basically, most individuals are already adapted to the conditions within the fundamental niche, and it takes longer just to establish populations outside these conditions. So rates of adaptation outside the fundamental niche are probably slower than extinction rates, but also dispersal and colonization to other conditions is less likely. However, there are ways in which the niche evolves, and of course it does. Niche can expand or shift, and these types of changes are what, in a sense, it's been called niche innovation, and it's what this paper by Holt and Gaines pointed out as niche evolution. However, we can also have retractions, right? And those could be caused by selective pressures or neutral pressures. So, Jonathan already pointed out some really cool papers that talk about niche conservatism and how it's a general pattern. I just wanted to point out here that in general we take it as an assumption or as a null hypothesis. It has been shown that at least climatic niche or broad scale conditions are conserved between putative sister taxa, and recently in 2024 there's a paper in which what simulations were used with virtual species to show that conservatism can promote allopathic speciation. The main point of this slide that I want to make is that niche conservatism is a key assumption of any exercise of ecological niche modeling, because when we are projecting models to other areas, like when we're working with invasive species, or when we want to understand potential past and future distributions, we are assuming niche conservatism. So we are assuming that climatic niche is not evolving. And in that sense, it's our tool for time travel, to understand past and future distributions of, or the lineages of our interest. So, today I'm going to talk about these kingfishers and toranthus, but my general aim is to, with this approach I'm going to present here, some preliminary results, is to estimate the degree of niche conservatism in these large-scale dimensions, mostly climate. And one way in which we're doing this is to compare ancestral niches to descendants, in contrast to previous analyses or how it's mostly done in which sister taxa are compared. For that, I've been working in UNM with these key features, the genus Toiramphus, which has 29 species, 93 subspecies, is a great speciator. They are distributed from the Red Sea towards French Polynesia, and they are quite diverse in terms of distribution. Some species are widespread, and some are only found in tiny, tiny islands in the Pacific. Here I'm summarizing some of the methods. I'm glad to talk more about this in detail later, but basically we took a bunch of occurrences from repositories. I analyzed here 2 environmental layers, temperature and precipitation. I did some estimations of accessible areas using packages like Grinnell and Place to This. I use a time calibrated phylogeny from collaborators that I will invite you to see Jenna's talk tomorrow. And we did marginal ancestral reconstructions to estimate ancestral niche ranges with the package Nicheable, and that's based on that paper from Hannah Owens in 2020. Quick shout out, if you want to know more about places, you should go to David's poster today. And as Chad already said, go to Jenna's talk if you want to learn more about Toiramphus. I'm happy to talk about Grinnell with whoever is interested later on. But I do want to point out some details about the ancestral reconstructions. So basically, when we have an accessible area estimated and we have occurrences of a species, we can classify the conditions in which they are present, right? Kind of estimating that convex curve of tolerances, in this case of temperature. And we bin range, or we categorize this range in little bins, and for each of those, we do an ancestral reconstruction. And here we are basically saying conditions can be suitable, or where the species is present, could be absent, or could be uncertain if they are outside of what is accessible for the species. So as I mentioned, that's how we do the ancestral niche reconstructions. And if we don't see change, we're basically saying there is niche conservatism. But change can be detected with this analysis as range retractions, expansions, or shifts. So with that, I'll show you some data. These are the character matrices of the 2 variables, and that's the general range in the bottom for the whole genus. But as you can see, there's a lot of overlap in thermal ranges between these subspecies, and ranges in precipitation are a little bit more variable in size, and there's less overlap. And what we found is that, so here in the tree, I'm pointing out in red the branches of the tree that show change or niche evolution, right? And what we found is that more changes were detected in the precipitation variable than in thermal ranges. But as you can see, changes are not equally distributed across the tree. We see a lot of changes in lineages with long branches, or that diversify earlier, like these species in the Philippines and in New Guinea, but also these widespread Parapyges species in Australia, Maclae, which is in Australia and the east coast of New Guinea, and also in more recent groups that are widespread, like Chloris is the only one that is in Africa and Asia, and Sordidus, which has become one of my favorite ones, and I'll show you why, which is in the northern coast of Australia. So a lot of changes in lineages with long branches and continental taxa, or taxa that are in large islands. Did I do that? I don't know. I'm not doing that. Okay. Also, we can see the changes as classified by retraction, expansions, and shift. And one cool result, I think, is that we detected more retractions than expansions of shifts, than innovation, which makes sense given that retractions can be caused by drift or other neutral processes, right? Right. It's there. Oh, okay. Now I just want to keep, because this is preliminary, we're still going through like all these little niche evolution events that we are detecting. I just want to show you or show, one case in this of what we have found. This is the complex sortus is composed by 3 subspecies, and Pilbara, the one that I'm going to focus now, they're in the west coast of Australia and showing the accessible area, and the records are in green, although they might be too small to see. What I want to show you is that the history of the niche range can be shown in this kind of plot, and basically each row is a node that takes you to Pilbara, and you see massive conservatism until 1.4 million years ago when it split and showed that niche shift. For an illustration, here are the other 2 members of that species, and those ones show conservatism with respect to the ancestral Toyramphus range. And that change can be seen in geography, and this map just shows that the species is present in the conditions to where it expand, right? And in a broader context, it's absent in the sites that hold conditions where the ancestor and the other subspecies of the complex are present. So I think it's a really cool result. We have other branches that show similar results. I just want to showcase that and say, yeah, it seems that niche evolution during Pleistocene like restricted this taxon to the driest part or the dry coast of Australia. So in summary, fairly conserved dimensions, but there are several niche changes, mostly retractions, and a lot of the changes are found in lineages that diversify early on continental lineages, and estimating the degree of niche conservatism in this phylogenetic framework can inform our interpretations or has informed our interpretations of diversification patterns. But I think there's a lot of that it's still need to be analyzed. So with that, I will thank you, and I will take any questions you might have. Yes, we can talk.
So there's been a lot of climate change, of course, in the past 10 million years. So how do you kind of account for changes in temperature in different localities through time?
Yeah, so definitely, like with this approach, we can project to those, if we have layers of those times, we can project back and see kind of like the range of analysis and how climate has changed. We haven't